Hi friends, meeting you after long days. So hope you all are doing very well. I was also quite busy so I couldn't record the videos for you. But this time I'll start with the previous year's papers of CSIR NET. So starting from 2015, we'll go up to the latest papers. First of all, we'll cover the part B portions of the previous year's papers starting from 2015. So today our topic of discussion will be June 2015 paper part B. So let's begin with the first question. This question is from Biochemistry. It says a 1% weight by volume solution of a sugar polymer is digested by an enzyme 20 microgram whose molecular weight is 2 5 times 0. The rate of monomer sugar molecular weight 400 liberated was determined to have a maximal initial velocity of 10 milligram formed per minute. The turnover number minute inverse will be. So first of all, whenever any numerical is given to us, we will write about the data which is given to us. Then we will solve our problem. So let's see. So Vmax given to us is 10 milligram per minute and E total, which we say as weight by molecular weight. Okay, we say E total is equals to weight by molecular weight. So what is the weight given to us is 20 microgram and molecular weight is 2 into 10 to the power 5. So we will write it here. This weight is 20 microgram by 2 into 10 to the power 5 is the molecular weight, and here Vmax, we denote Vmax as what? Mole per minute. And how do we write mole? As 10, which is the milligram, by mole is by molecular weight. So molecular weight here given is 400. Okay, by, okay, wait. So this is Vmax. Okay, now we will convert the minute into gram. So 10 milligram into gram is 10 into 10 to the power minus 3 by 400. This is now into grams, is equals to 2.5 into 10 to the power minus 5 mole per minute. Mole per minute. Okay, next if we solve this, it is 20 microgram. If we convert this into gram is 10 to the power minus 6. If we go from milligram to gram, it is 10 to the power minus 3. We'll multiply with 10 to the power minus 3. If we have to go from microgram to gram, we will multiply with 10 to the power minus 6 into 2 into 10 to the power 5 is equals to, when we will solve this, 2 got cancelled out, and here this 10 will go above. Okay, so it will be 10 into 10 to the power minus 6 into 10 to the power minus 5. So if we solve this, it will be 10 to the power minus 10. Got it? Now what we have to find out is turnover number. Turnover number is what? Kcat. We denote it as Kcat, and the formula for Kcat is Vmax by E total. Okay, so is equals to Vmax, we found out it is 2.5 into 10 to the power minus 5, and E total is what? 2.5, sorry, it is 10 to the power minus 10. Okay, and what was the unit? The first unit was gram per minute and the second was only gram. So gram got cancelled out, and what we get is 2.5 into 10 to the power 5 minute inverse is our answer. So answer 4 will be the correct option. Answer 4. I hope this is clear. Coming to the next question. This is also from biochemistry. Number 22. In an alpha helical polypeptide, the backbone hydrogen bonds are between. So, how the backbone hydrogen bonds are formed in alpha, beta, gamma, pi, this we have to memorize. Okay, this is very important. So let's see. So the backbone between the carbonyl group and the NH group for alpha helix is N plus 4. Okay, simultaneously you have to remember for the beta helix also, gamma and pi helix also. For beta it is N plus 3, for gamma it is N plus 2, and for pi it is N plus 5. Got it? So also you have to remember about some properties of alpha helix, like say for example amino acids found in alpha helix are like malic. You can use the code word, you can learn by this way. Okay, and there are several other properties of alpha helix. It's like the pitch is 5.4 per turn, is 3.6 residues, and so on. So guys, what will be our answer? Our answer will be, if it's alpha helix, it will be CO, carbonyl of N, and NH of N plus 4. Answer 3. Third question is, this is also from biochemistry. 23. Following are 3 single-stranded DNA sequences that form secondary structure. 3 sequences are given. This is going to form the secondary structure. Based on their stability, which one is correct? So first of all, we need to know that the ones having the more GC content are more stable. So if we have to say how to solve this, so Tm, which is the melting point, is directly proportional to the stability. Okay, and we find out the Tm by the formula 4 into G plus C plus 2 into A plus T. Got it? Now let's calculate about the number of GC and AT in all of these 3 strands. So let's first find out about the number of AT and GC in all these 3 strands. So first of all, AT in the first strand is equals to 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 ATs are there. And how many GC? 1, 1, 2, 3, 4, 5. 5 GCs are there. GC is equals to 5. Let's find out for the second. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 80 are there. And GC is how much? 1, 2, 3, 4, 5. 5 GCs are there. For third one, 1, 2, 3, 4 and 5 ATs are there. And how many GCs are there? 1, 2, 3, 4, 5, 6, 7, 8, 9 GCs are there. Now what we are going to do, we are going to find the Tm for each of these strands. So for number 1, it was 980 and 5 GC. For number 2, it was 1180 and 5 GC. For number 3, it was 580 and 9 GC. Okay, how to find the Tm? Formula is given. Tm is equals to 4 into G plus C plus 2 into A plus T. So 4 into G plus C is 5 plus 2 into A plus T is 9. Okay, is equals to 5 fours are 20, 9 twos are 18 is equals to 38. Okay. Second is Tm is equals to, I write in this way, 4 into A plus T, which is 11. Sorry, 4 into G plus C, which is 5, plus 2 into A plus T, which is 11, is equals to 20 plus 22 is equals to 42. Third is 4 into G plus C is equals to 4 into 9, plus 2 into A plus T is equals to 5. So 36 plus 10 is equals to 46. So now we have to arrange that which one is more stable. So the one having higher Tm, that is more stable. Okay, so here C is more stable than B than A. So this is the order of the stability. Now I found this as the correct method, and this option is not given. The option given to us is CAB, which is the correct option shown by CSIR. So if you find out, if you have any input, then you can please write it in the comment box. Thank you. According to me, C will be greater than B than greater than A. If you have any inputs on this, then please write it on the comment box. Next question is again from biochemistry. Which one of the following enzyme is not a part of pyruvate dehydrogenase enzyme? Which one of them is not? So whenever a not term is mentioned, you have to select your answer very carefully, right? So pyruvate dehydrogenase complex enzyme. First of all, what is the role of this enzyme? Now this enzyme, pyruvate decarboxylase, what it does, it catalyzes the oxidative decarboxylation of pyruvate. Okay, it catalyzes the oxidative decarboxylation of pyruvate, and this enzyme and this complex, this enzyme forms a complex. This enzyme forms a complex, and this complex consists of three enzymes. Consists of three enzymes. Okay, what are the three enzymes? Number one, it is pyruvate dehydrogenase. Number two, dihydrolipoyl lipoyl transacetylase and dihydrolipoyl dehydrogenase. These are called as E1, E2 and E3 enzymes. And these enzymes also require coenzymes. What are the coenzymes they require? Pyruvate dehydrogenase requires TPP. Dihydrolipoyl transacetylase require lipoic acid, and coenzyme A. And the last one, dihydrolipoyl dehydrogenase, it require NAD plus and NAD. Sorry, FAD. Okay, and there are two cofactors. Okay, there are two cofactors. Sorry, there are two ions which inhibit E1. What are those? Arsenite and mercuric ions inhibit E1. Okay, now let's go back to our question. Which one is not the part? So pyruvate dehydrogenase dihydrolipoyl transferase is the part, dehydrogenase is the part, but oxidase is not involved here. Okay, so E1 is dehydrogenase, E2 is acetyl transferase, which is lipoyl transferase, and E3 is dihydrolipoyl dehydrogenase. Right? Hope this is clear to you. Now here we are done with the biochemistry part. We will start with the part B of cell biology part in the next video. Thank you.